Hello everyone, my name is Farhad Razi, and in this video I'm going to walk you through some of our recent results. During the deep phases of sleep, we do not normally wake up by a thunder, but we nevertheless notice it when awake. Extracellular cortical recordings in the primary auditory cortex of rodents show that an increase in firing rate in response to pure tones during non-REM sleep is comparable to those evoked during wakefulness. This result challenges the hypothesis that during sleep cortical responses are weakened through thalamic gating. An alternative explanation comes from the observation that the spatiotemporal spread of the evoked activity by transcranial magnetic stimulation in humans is reduced during non-REM sleep as compared to the wider propagation to other cortical regions during wakefulness. Moreover, in rodents, neural activity recorded in cortical areas higher up the sensory processing stream shows significant differences between sleep and wakefulness as opposed to what happens in primary auditory cortex. These results indicate that the responses to auditory stimuli shift from a local to a wider propagation pattern from non-REM sleep to wakefulness. One possible neural correlate of decreased sensory responsiveness in the non-primary cortex during non-REM sleep could be the downscaling of the conductance of excitatory synapses to compensate the net increase in strength during wakefulness caused by synaptic plasticity, a hypothesis known as synaptic homeostasis hypothesis. We have used the computational neural mass model describing two cortical columns, each consisting of one pyramidal and one inhibitory population. The pyramidal population of one of the columns is subject to an external input modeled as a brief square pulse. We quantified the propagation of this excitatory input to the unperturbed column as a function of the coupling between columns, which is only excitatory. To do so, we have computed an independent t-test comparing the post-stimulus versus pre-stimulus firing rate signals of the unperturbed column at every time point and formed clusters of consecutive time points above a critical t-value, called t-cluster statistic. The size of area and duration of the first cluster quantify the propagated information to the unperturbed column. Following the synaptic homeostasis hypothesis proposed by Giulio Tononi and Chiara Cirelli, upscaling of the excitatory conductances G-AMPA allows us to place the model from non-REM sleep into wakefulness. Simulated signals such as the local field potential or LFP, population firing rate, and population membrane potential show large amplitude fluctuations during non-REM sleep, whereas during wakefulness the fluctuations exhibit low amplitude components. The distribution of LFP signals shifts from a bimodal to a unimodal distribution from non-REM sleep to wakefulness. Moreover, the ratio of high over low frequency content of the LFP signals is larger during wakefulness than non-REM sleep. In agreement with the experimental results from auditory cortex, the endogenous dynamics during non-REM sleep produces a comparable, even higher response to the external input to the ones during wakefulness in the perturbed column. During non-REM sleep and wakefulness, the average evoked responses in all signal types exhibit significant deflections denoted by red bars on top of each graph. On the contrary, the evoked responses in the unperturbed column are not significantly different from the spontaneous activity neither during non-REM sleep nor during wakefulness. we have found that the ratio of the inter to intra conductance of cortical excitatory synapses, called beta, should rise to facilitate information propagation to the unperturbed column during wakefulness. Keeping beta equal to 1 does not trigger a significant T-cluster statistic in the unperturbed column even when potentiating G-AMPA from 2 to 10. Importantly, a selective increase of G-AMPA for the inter-excitatory connections over the intra-excitatory connections quantified by beta enlarges the area and duration of the T-cluster statistic. To our knowledge, this is the first mathematical model that reconciles the two important findings of sleep research: synaptic homeostasis and the simultaneous emergence of slow rhythms as the brain enters the deep phases of sleep. Our results suggest that potentiation of excitatory conductances during wakefulness should occur preferentially between distinct networks over local and recurrent connections, a spatial-selective synaptic homeostasis policy. Thanks for watching.